morning to all. I am Ms.C.Petchiammal, Assistant Professor, the Standard Fireworks Rajaratnam College for Women, Sivakasi. So today we are going to see about the topic is liquid chromatography. So learning objective. So this includes principle and types of liquid chromatography, procedure of liquid chromatography and advantage and application of liquid chromatography. So what is liquid chromatography? So liquid chromatography is a technique used to separate, identify and quantify the components of a mixture. So it is a differential interaction of substance between a stationary space and a mobile space. So stationary space is usually a solid or gel components and mobile space is a liquid components. So the components of the mixture travel through the system at different rates enabling their separation. So next, the principle of liquid chromatography. So the principle of liquid chromatography includes stationary space, mobile space and separation mechanism. So the stationary space is typically a solid or coated material packed in a column. So it interacts with the components based on chemical affinity, size or polarity. So mobile phase is a liquid that moves through the stationary space. It can be a single solvent or a mixture. So the mixture example water, methanol and etc. So it carries the sample through the column. So next is the separation mechanism. So these components in the mixture interact differently with their stationary and mobile space, leading to varying travel times. So travel means retention times. The type of interaction depends on factors like polarity, size and ionic changes. So next, the types of liquid chromatography. So it includes partition chromatography, adsorption chromatography, ion exchange, size exclusion and affinity chromatography. So the partition chromatography, so the separation is based on the partitioning of components between two immiscible phases. So as example aqueous or organic spaces. Next, the absorption chromatography. So components adhere differently to the solid stationary space based on surface interaction. So third one is ion exchange chromatography. So chromatography separate ionic components based on their affinity to charged groups in the stationary space. And then size exclusion chromatography, it means SEC. So it separates molecules by size and larger molecule elute faster as they bypass smaller pores in the stationary space. So last one is affinity chromatography. It uses specific biological interactions, example antigen antibody interaction to selectively bind target molecules. So next is the procedure of liquid chromatography. So what are procedure for liquid chromatography involve the following steps. So first one, inject the sample. A small volume of the sample is injected into the column using a injector. So pass the mobile phase through the column. A liquid solvent called the mobile space, say pass through the column at a constant flow rate. Separate the components. The sample components separate as they pass through the column due to difference in how they interact with the stationary space. So next, detect the separated components. So this detector monitor the separated components as they emerge from the column. Finally, the produce a chromatogram. So what is produce a chromatogram? Chromatogram means the detector produces a voltage response over time. So it is called a chromatogram. The time it takes for each peak to emerge identify the sample components and the peak area represent is quantity. So next, this is a basic LC systems consist of the instrumentation. So A solvent inlet filter, B means pump, C inline solvent filter, D injection valve, E petroleum filter, F column, G detector, H means recorder, I back pressure regulator, J waste reservoir. This is overall instrumentation. So next, advantage of liquid chromatography. So it is high sensitive for detecting and quantifying components and applicable to wide range of substances, means polar, non-polar and ionic substance. Flexibility in mobile and stationary space allow for tailored separation. Compatible with complex biological mixture. So next, application of liquid chromatography. So many field is application of liquid chromatography, so pharmaceutical, environmental monitoring, food analysis, biochemistry and chemical research. Pharmaceutical industry is a drug purity testing and stability studies, so whatever it is. So environmental monitoring, it means detection of pollution in water, soil and air sample. So food analysis, so it identifying additives, contaminants and the nutritional components that are involved. So biochemistry is involved is protein purification, DNA analysis and metabolic, so etc. So last one's chemical research is synthesizing and separating new components. So that's all liquid chromatography overall procedure, application and advantage. Thank you for watching this video.